From the editors of Biological Psychiatry and the Society of Biological Psychiatry, this is the Biological Psychiatry podcast series. I'm Tamara Gore, and today I'm talking with Dr. Deborah Bangasser. Welcome.
Thank you for having me.
Great. Could you tell me a little bit about yourself first?
Yeah, sure. I am an associate professor at Temple University in the Department of Psychology, and I also run the neuroscience program in the College of Liberal Arts.
That's great. We're here to talk a little bit more about the paper that just came out in our journal. And I was hoping to start by you telling me a little bit more about the known relationship between stress and psychiatric disorders.
Sure. So it might be easy to think of a disorder like post-traumatic stress disorder or PTSD that's actually precipitated by a traumatic event. But unchecked stress can exacerbate the symptoms of many psychiatric disorders, ranging from attention deficit hyperactivity disorder to depression. So it's really important to understand the consequences of unchecked stress if we want to improve treatments for these disorders.
Got it. How is stress known to impact attention?
So anecdotally, we can probably think of examples of stress impairing our ability to attend to something. You know, sometime when we were really stressed out, it might have been hard to concentrate. And there's studies that in humans, life stress, so people that have had a lot of life stress, tend to have more impaired attention. But the effect of repeated stress on attention hasn't really been extensively explored. And in our lab, we do that in a rat model because it's easier to manipulate the stress and more ethical in certain aspects. And I think we were the first lab to look at the effects of repeated stress in adult animals on sustained attention. And that's our ability to attend to things in our environment that are coming in kind of an intermittent or unpredictable way.
And so tell me more about what new findings your study contributes to our understanding of this process.
Yeah, so we studied the way this repeated stressor, so they got a stressor every day for 6 days, and we saw how it affected the sustained attention task, and we saw that this stressor impaired attention in both male and female rats. We know that this task requires cholinergic neurons in the nucleus basalis of Meinhardt, which I'm going to call the basal nucleus, and that the release of acetylcholine into the prefrontal cortex from these neurons is really critical for sustained attention. And so the advantage of using an animal model is that we could kind of start to determine, well, how is stress affecting these cholinergic neurons? And we knew from other studies that repeated or chronic stress can actually change the morphology or shape of cholinergic neurons, often affecting the dendrites. And remember, that's a part of the neuron that's processing input. So if there's changes in the dendritic morphology, it could affect how inputs into that system are being processed. Okay. And so we wanted to look at whether this repeated stressor was affecting dendritic morphology and cholinergic neurons. But to do that, we had to develop a new approach to visualize only cholinergic neurons in the basal nucleus. And we did this by taking advantage of a Cree chat rat that was developed and a virus that reports in the presence of Cree, so we could visualize these cholinergic neurons. And then what we did is we said, well, how does this repeated stressor affect these neurons? And it turns out that in both males and females, this stressor caused the dendrites to get longer and more complex. So it actually induced dendritic hypertrophy.
Wow.
And I think actually it's very interesting. We also saw a baseline sex differences where these neurons are longer and more complex in male rats and female rats to begin with. And no one had really looked at both sexes before because it was hard to look at these neurons. So that was also an interesting kind of side finding, but the stressor is causing this hypertrophy in both males and females. And these dendrites are under a lot of inhibitory control. So what we think might be going on is that they're getting more inhibitory input, making it harder for them to appropriately release acetylcholine in the prefrontal cortex. So that was our next hypothesis, and we tested that, and we saw that that was the case in males. So males release less acetylcholine into the prefrontal cortex following stress, and we think it might be because their dendrites are changing shape. Interestingly, though, this is where we saw a sex difference. We did not see this effect in a decrease in acetylcholine release in the prefrontal cortex in females. So we're not sure what's changing in females to cause the behavior, and so we have more work to do there.
Very interesting and very exciting. If this were a perfect world, what psychiatric treatments would be developed in light of your findings? How do you think this might impact psychiatric treatment?
Well, so as I mentioned that there are a lot of disorders that have stress as a feature and attention as a deficit. We know that for other types of disorders, acetylcholinesterase inhibitors, which increase the production of acetylcholine basically, are used. So for Alzheimer's disease, those are a treatment. And actually in Alzheimer's disease patients, these drugs tend to improve their attention symptoms. So it could be that some of these drugs could be repurposed for other disorders with stress and attention. Maybe given our sex difference, this might work better in males, we're not sure.
Really exciting.
Another thing
would be a perfect world to get started thinking about.
Could be, yeah. It could be that we could take something we already know. Another thing we did in our studies is we used RNA sequencing to look at the gene transcription that changed in the basal nucleus following the stressor to get more at the molecular underpinnings. And we have some interesting gene changes that might be driving some of the changes in plasticity that we're seeing in these cholinergic neurons. So perhaps if we can follow up on those, we might also get insights into some novel treatments that could be used to improve attention during stressful times.
It's certainly very intriguing. If I forced you to hazard a guess, what would you think your study might suggest might be happening to those of us experiencing the stress of COVID-19 and the resulting shutdown and other changes in our way of life? What do you think might be happening in my nucleus?
Well, so first I'll say kind of a broad answer, and then I'll do like a more maybe neuro answer. But I do think studying this topic, and certainly these data also support the idea that we should not be surprised that even when we find the time to work, we might be having a harder time concentrating than we normally would, right? Because this is a stressful time and stress is disrupting our attention. In terms of molecular changes in the brain, it's possible that the things we see in rats are also occurring in humans. We don't know that for sure, but it could be that there's some changes in our cholinergic neurons. Now, we don't know this from any of our data, but changes in morphology of neurons in many other circuits in the brain, like the hippocampus, for example, tend to go away after the stress is removed. So if we are experiencing some stress-induced changes in our cholinergic system, I would hope that those would recover. You know, our brains are pretty resilient. Once this pandemic is over, hopefully we can get back to normal.
I deeply hope so. And I've really enjoyed talking to you this afternoon. Thank you so much for taking the time.
Thank you so much for this opportunity. It was fun to talk about the data.
Absolutely. Thanks.